Okay, good morning, everyone. And so welcome to this school. So thanks to Abhishek and Sanjib for giving me this opportunity to present a bunch of ideas in pattern formation. So I'll give a bunch of lectures on broad aspects of pattern formation in biology. I mean, the very word biology must scare off many physicists as usual. But my idea is not to go into the details of biological systems and their jargon, but to give you a feel for looking at living systems as basically condensed matter systems describable by statistical physics methods. So that's the approach that I want to take. So feel free to interrupt anytime. My idea for the lecture today is that I will use the slides to show you videos and pictures and images, mostly videos and pictures and images for like 40-45 minutes roughly. And then towards the later half, I'll go to the blackboard and we'll start setting up a set of nice mathematical ideas to understand how to describe these patterns in more quantitative detail. So if many things don't make sense, the equations that I flashed through right now, just bear with me. I'll work through most of the details in the subsequent lectures. Okay, pattern formation, I mean, as most of us know, is something that you see in systems typically driven out of equilibrium. So you have some field, maybe a density field, maybe a flow field, whatever it be, and there is some non-trivial spatio-temporal aspect of the field. So for example, if you do a kitchen experiment where you put a liquid in a pan and just heat it at the bottom, beyond a certain temperature difference between the top and the bottom, you see these natural swirl patterns that come up. These are the classic Rayleigh-Bénard convection patterns, which I presume most of you would have heard. The physics underlying these things is basically fluid mechanics, Navier-Stokes equations, coupled with some heat transport. And beyond a certain temperature difference, it so turns out that the conduction state is unstable. And so you set up these nice convection rolls coming up. A similar physics governs pattern formation even much larger scales. I mean, this is a weather pattern seen from the Space Telescope or the International Space Center. And to scale, I mean, if you see this black dot here, that's an island somewhere in the Pacific Ocean. So these are scales which are thousands of kilometers wide. And so this is classical von Karman vortex street patterns that you see. All right. That apart, I mean, there are patterns in the natural world which are very, very ubiquitous. I mean, starting from plants and trees to cracks in mud piles or the meandering of rivers. There's very, very, very wide variety of patterns. So most of the living world, most of the interesting parts of the natural world can be classified as basically some sort of a patterns, right? So classical examples of pattern formation in biology typically come from looking at pigmentation patterns in organisms. Snakes are nice because they're naturally one-dimensional systems, so you can do nice Statnecki models. But they don't show simple patterns. There are snakes with various kinds of one-dimensional patterns. So there are patterns which go along the length of this body, perpendicular to it, maybe spots, maybe stripes, maybe a mixture of them. There's a huge variety of them. So it's always a curious thing to understand why these patterns. I mean, what's the use? Maybe you can cook up something about to say, oh, if it was not that, it would have been eaten up. It's a nice camouflage. Sure, but how does those patterns form? I mean, it's a natural curiosity to understand, right? A similar thing comes up in animal coat patterns, again, something that all of us should have wondered when looking at these things as kids. Why does the leopard have its spots? Okay, there's an evolutionary answer to that probably. But there's what one is trying to look at is more of a physical answer. How do we understand these kinds of pattern formation in living systems? Fish skin patterns, another classical example, right? So there's a wild variety of all these things. Why have them? I mean, couldn't you just be fine with just one kind of fish pattern? Why do the creatures have these kinds of patterns? All right. These are patterns at the scale of organisms, large scale pigmentation patterns, which typically come from certain spatio-temporal rearrangement of certain color-producing cells, pigment cells. These kind of patterns are seen even at the single cell level. I mean, for instance, the most dramatic and remarkable thing about living matter is the fact that it replicates. Well, if I take this pointer and I put it there and I give it any amount of energy input, I'm sure it will not make a copy of itself, right? I mean, but a cell does that. And very remarkably, very robustly, and it's happening in every one of our bodies right away. So it's a fascinating thing to even ask what is the statistical physics of such processes? I mean, you see a cell with the DNA marked in red, and these green colored things are certain polymeric fibers. We'll discuss a little bit about them towards the second or third lecture. And the process essentially turns out to be that there's a condensation of these red things. So that's the process where the DNA is duplicated. And after that, these green-like stripes, they pull apart the chromosomes into the two daughter cells. This is typically of the order of few tens of microns in size, and this roughly takes about an hour or so, half an hour to 1 hour. So how is it that this pattern of DNA condensation is happening? How is it that what are the forces that are coming up to pull apart this, and how do these things happen? One is at the cell level, cell division level, and other kinds of patterns that come, which are very important in establishing axes in organisms, is polarity patterns. So you see a cell, which is from a zygote of a creature called C. elegans, a standard organism which is used. And what you're looking at is the three-dimensional cell, and you're looking at one equatorial plane. Okay, so it's a confocal imaging. And so what you see is a typical surface pattern, which is initially homogeneously red to begin with. And something happens and a red-blue pattern automatically develops on the surface of the cell. So this red-blue pattern, turns out living systems use such chemical patterns, typically some sort of a segregation pattern, to define an axis. So you can obviously draw a vector from the red side to the blue side, right? So that's defining a coordinate system for you. So this turns out to be a serious problem in biology of developing coordinate systems. Just to see the fascination of how these things happen. So one is at the cell level, how a single cell divides, how a single cell polarizes. But then if you go up and ask, how does a multicellular organism develop, starting from an embryo, which is basically a single cell, almost a single cell. And there's a single cell here. There's a bunch of yolk cells, which is basically food for the creature. And it's more or less an isolated system. I mean, there's a temperature and pH exchange. But apart from that, you don't do any external interventions to the system. You watch it under a microscope, and you see a remarkable pattern of development coming up. So this is a developmental movie of a zebrafish embryo. So you see the cells divide into tiny, tiny pieces, and then they start reorganizing themselves in some particular fashion. And you see time running somewhere there. And at some point of time, you start seeing the appearance of very nice periodic patterns here. That's your backbone. It's the typical vertebrate pattern that comes up in all of us. You see some pigmentation patterns coming up. At some point of time, you should have seen an eye-like structure coming up. So it's a fascinating thing to see that an organism, that a system which is left to itself, has this ability to self-organize. And so to generate patterns. Patterns in what? Patterns in, obviously, cell sizes, cell shapes, cell positions, pigmentation patterns, coloring patterns, but probably more importantly, patterns in shape, patterns in geometry. So not something which is completely a spherical object, but something which develops a head, a tail, a limb, a fin. So these are also patterns, right? So it's a fascinating problem to understand. Why is it that these patterns come up? I'm not saying we understand them. We're just opening our eyes to these kinds of physical processes here. Another example, something that you should have all seen, I mean, at least this one, not this level. So if you keep a banana open, you see all these tiny insects, they come and sit on the banana there. That's black insects, and maybe you push them away by annoying. These are fruit flies, tiny organisms. It's a standard developmental organism because we know the genetics of this creature very, very well. And so it's useful to look at how it develops. So this is basically an egg of the creature, and you put it on a microscope and you watch it. And again, it's enclosed in an eggshell, so therefore there's no energy input going on. There's some pH, some temperature, and you see a remarkable transformation in the gooey contents of this creature, whereby a bunch of cells divide. They divide many, many times. They rearrange. At some point of time, you'll see something shaped like a gut coming up there. And at a later point, the whole fly forms and the fly flies away. So it's an absolutely fascinating thing. I mean, how is it that these things work? I mean, I thought entropy should maximize in equilibrium and everything should be flat. But now you have a system which is organizing spontaneously. So it's natural to ask, what's the physics of these questions? During the course of its development, this fly, it so turns out, is this visible at the back? Okay. For me, the contrast is quite bad. So during the course of its development, it so turns out that this fly, the very fly that I showed you, develops wing patterns. I'm sure if you played around with the fly during a curious moment of probably killing it, you do see wings. Wings have veins, and there's a particular shape of the wing. And so during this course, you actually see this very fantastic formation of a wing-like pattern. So you see a mass of cells to begin with, which seem to flow inward. The flows are spatially dependent. At some point of time, you see the formation of a very nice wing-like pattern, and behold, the veins are also forming up there. This is almost a single layer of cells. So this is just a few tens of microns thick and several hundred microns wide. And this is so many R's after some nice point, which you reckon as t equal to 0. So all this is happening inside the eggshell. So while the creature is developing. Okay, so one way to think about such models is to write down maybe some simple lattice models or models where you can have the cells modeled as polygons, and you write down some simple elasticine models, for instance, where you say there's some cost for deforming the cell, there's some cost for changing the perimeter, and you put in some ad hoc things by which try to mimic the active forces sitting there. So such models have been written a lot and studied a lot. I only put this slide to say that I will not be talking about these kinds of models. But just going up and ask, what are the kinds of problems? What are the physics problems here? So one is in looking at multicellular development. One set of problems is how do you make different kinds of cells? I mean, how does a stem cell become a red blood cell or a neuron or a fly but this is more of a question of biochemistry, which genes are expressed there. And so we need physics aspects there, or the kind of things that one would be interested in. The other problem is morphogenesis, how to shape up the creature. So problems like axis establishment or segmentation, these are genuinely problems in pattern formation. And these are the kind of problems that I'll give a feel for. So think about the following. A typical creature starts out more or less spherical, maybe an ellipsoid. How does it know its head and tail, front and back, left and right? So if you don't know that, you don't have a coordinate system. You don't know where to place which organ. So how do you measure distances? How do you measure orientations? So this problem is really one of doing an axis establishment. The other problem is once you've done some axis establishment, how to chop up the creature, how to make its head, tail, develop a wing, and so on. So here are two examples of the same ideas of pattern formation. So this is the fruit fly, Drosophila again. And if you look at the early divisions, so this is the same movie seen from two different orientations, you see very nice stripe-like patterns that will come up towards the end of the movie. This is very early in embryogenesis. And so it's been a puzzle to understand how these stripes come, what sets the periodicity of the stripes, how accurately the stripes are positioned. And the importance of the stripes appears because these stripes that you see at the very early stages of development are the ones that correspond to organs in the adult. So there's a very nice one-to-one mapping between them. You should use the mic if you don't mind.
See, in that thing, once your already the starting point was like a ellipsoid, right?
Yes, yes.
So was there a sphere kind of thing?
No, no,
no. It's already the
it starts from an ellipsoid.
So it's already axis is established, but you don't know head and tail.
You don't know head and tail. In fact, it's not even a process of self-organization. I was coming to that. So it turns out in biology, you seldom get pattern formation by instabilities. That's very bad because it's uncontrolled. You don't know when the pattern. So there's some sort of signals that the mother has laid at the beginning, which sort of defines this, but the subsequent pattern formation is self-organized. So feel free to ask questions if things don't make sense. Okay, so this is one example, the problem of segmentation. How do you chop up the creature into different parts? The other one is one of axis establishment, and this is something I showed you in a moment ago. So this is basically the development of a C. elegans embryo, and what you see is the very first cell division, and you see the very first cell division, the two daughter cells are different. So some chemical, it doesn't matter what the name is, went to one of the daughter cells and not to the other preferentially. So therefore, one of the daughter cells is different in fate because it's got a different set of chemicals. So the questions of how does it even know how to establish this kind of a pattern? So this pattern, you remember I showed you the surface pattern, guides this pattern. So this pattern forms the red and blue pattern on the surface of the cell. The interface between the red and the blue is where this division happens. And this light blue really goes to the
What is that red and blue thing?
These are some proteins. You don't need to know the names of the proteins. Okay. So see, even fundamental problems like how do you know your head from your tail is really related to how do I form a pattern which can distinguish a head-like region from a tail-like region. So therefore, it's impediment that we understand how do these patterns form. So one is to say at a qualitative level and make some theories probably which will give you some nice patterns. But experiments from the past few decades have become so good in biology that you can actually measure things very, very quantitatively. And so maybe in the second or third lecture, I'll show you some data from my own work, which measures this pattern very, very quantitatively in terms of number of molecules per micrometer square. And we'll write down a theory based on some simple reaction diffusion and active patterns and show that these things quantitatively can be captured by that. So therefore, it becomes a problem of really good physical measurements and developing good physical theories to explain these patterns.
So in this case, like the code for the head and tail were already there, or it just like spontaneously
So there's a lot of, so the question was, is there a code which is already there for the head and tail, or is this a spontaneously organized pattern? So it turns out there's a mixture of things. So there are the homogeneous red state and the homogeneous blue state, and the pattern state seem to be stable at the same time. And what you're seeing is a transition from one self-organized state to another self-organized state driven by a signal. I'll work out the details of this sometime later. All right. One is to look at a single cell, multiple cells, and look at how multicellular organisms develop. But you can also put a bunch of cells together and look at the collective dynamics of such systems. So this is bacteria. And you put a bunch of them in some petri dish and you're just observing their motion. You see these fantastic patterns where there are rod-like structures moving around in particular directions. But remember, these are, okay, I don't know why the movie is not playing. Okay, so these are objects which are elongated. So therefore, there are steric interactions, and the steric interactions are not isotropic. So therefore, in addition to certain patterns in densities, bacteria more here, bacteria less there, you should also expect patterns in orientations. Bacteria more aligned here, bacteria less aligned there. So therefore, the complexity of the problem goes up. So rather than thinking of simple density fields, maybe you should start thinking of, let's say, vectorial fields, polarity fields. It becomes much more evident if you look at birds. I'm sure you should see this, you would have seen this somewhere. You can also see this in ICTS at some point of time. Each dot there is a starling, is a bird. And so when you look at huge number, tens of hundreds of thousands of birds, and at that scale, you can't really say it's a bird. I mean, it's really a fluid. And so therefore, the fluid is not just sitting like there with a droplet statically. It's moving around, and you see huge density variations. There are patterns forming there. Obviously, they're going somewhere. I don't know what they're doing. They're trying to find food. They're going home. We don't know that. But the kind of patterns that come up at this large scale level is fascinating. So one could just ask, can I do some fluid physics of these things? Is it like a droplet of water, or is it not like a droplet of water? We don't know. I mean, so one of the things that one tries to do at these levels is not to worry about the details of how each bird sees each bird, does it like it or not, does it color, but to write down what's called a hydrodynamic theory, fluid dynamics, elasticity. So that's the approach that I've also taken in my lectures. And such things have been done from the past 2 decades. And so what you think about a bird, each bird is basically an arrow, a polarity vector sitting there. And each bird likes to align with its neighbors. Because I mean, surely if your bird is going like that, that's not good, it'll crash onto each other. So better align with each other. And so some simple physics of like ferromagnetic alignment in a Heisenberg model, if you just put in that, and you write down what is the hydrodynamic description for these things, turns out that this is quite good. So such theories have been developed. I don't want you to understand much of this, just basically the idea that there is a density field and there is a vector field, which is like a velocity field and also an order parameter field. And there's some physics. I'll try to describe some parts of this. And so it turns out that the predictions of this kind of active hydrodynamic theory can quantitatively capture these kinds of phenomena. So you can ask questions like, How does the structure factor look like as a function of the wave number? How do the giant number fluctuations look like as you increase the box size? So these are predictions that can be made from a theory of this sort, which has been tested in experiments. So the fantastic experimental work done by people in Rome, they actually had, I don't remember, maybe hundreds of cameras now, put up at some point, they captured the birds. And so by doing very smart image analysis and machine learning algorithms, you could figure out where the bird is, what the orientation of the bird is. And so therefore you could get huge amount of data and so do lots of static. The thing to note when constructing such theories is these are systems which are genuinely out of equilibrium. Okay, so in the sense that they break detailed balance. So there is a flux of energy. At this scale, it's basically the food the creatures eat. So if you don't starve them, they'll die and they don't do this dance. So you need to supply energy. The other essential ingredient for these kinds of non-equilibrium systems is that they're active in the sense that the consumption and dissipation of energy happens at the level of the individual units. So that's all is basically the essential ingredient. The rest is a bunch of smart math to write down. And if you use those things, you can write down a general theory like this. So there is a part of this, some of you might recognize, it's something like the Landau-Ginzburg theory for magnets. So a strange mixture of magnet and fluid seems to describe this. Now, not just birds, you can also look at fish schooling. I don't remember the credit for this video. And fish schooling flocks. Sorry, what am I saying? Schools of fish have also been described very, very quantitatively as active materials, with the fish being a polar order parameter. Something that's very funny, and I occurred on Twitter yesterday, even sheep, for instance, right? So this is sheep being herded by dogs somewhere in the UK. There's a very fantastic aerial shot of this. And so if you really can't distinguish each one of the sheep, it's basically some polar order parameter. At some point of time, you'll be able to see some defects as well coming up there. There's one shot where it really basically looks like there is a fluid which is being pushed through a small orifice, and you really see a magnificent fluid-like behavior coming up. Okay, there's some vortex setting there. Okay. So the point that I was, okay, that's the picture. So you can really think about this like a fluid that's being pushed. So it goes out, and look at the way it expands out once it comes out. Okay. What are the patterns? I mean, so typically these are patterns in space, spatiotemporal, spatial heterogeneities, maybe intensity fields, maybe in some concentration fields of certain chemicals, maybe in time as well. And they typically break spatiotemporal symmetries. So that means you had a system which was homogeneous in space, unpatterned state, and you break that translational symmetry, and so pattern ensures. But in what? I mean, like I was saying, you could have things in scalars, vectors, tensors. So you could have densities in concentrations. You could have flow fields, alignment fields, like polarity fields. You could have stress fields or shape fields and orientation fields without a particular axis. So some of them we will work out, some of them in scalars and vectors will work out some of these patterns. So there are patterns like this, and one would like to write down a generic theory for understanding such pattern formation. So there are patterns in space-time and patterns in scalars. So what? I mean, why should it matter? What is the use in biology? I mean, do living things use them for something, or is it incidental? So this is a fascinating video that I wanted to show you, and it's very, very useful to think that how much is that we don't understand in living systems. So for people online as well, this is a nice exercise. I guess everybody should participate. There is an octopus in this image. So if anybody can get it, let's see. Take a guess. Yes. So there's an octopus in this image, and you can take a guess. Maybe you can say it's buried under the sand. Maybe it's behind the bush. I don't know what that is. So take a guess. Try to think about it. So anybody? Nobody? Top left. So somewhere here. Okay. Top left corner. Okay. Somebody else also says that. Okay. That's one thing. Anything else? bottom center here or behind the bush? Huh? Here. To the right, here, this one. Okay. Anything else? What? Huh. Bottom left, center, all right. I mean, if you have seen this video, please don't answer. Maybe the center, okay. Changing color, sure. I mean, that's the pattern that we are going to see. But the remarkable pattern will come up in a second. Central bush part, okay. Bottom left, okay. Left side of bush, okay, fine. All right, so let's go in. So this is a video taken by Roger Hanlon from the Marine Biological Laboratory. And so apparently he was swimming one day in the ocean there, and he went along and he was filming there, and there was the bush, and maybe there's something behind the bush, and you go around and you go close by, and oh my God, the bush is the octopus. Now that is bewildering pattern formation. So the movie will show you in a second about how the transformation happened, and it'll actually show you in reverse, and you'll see something shocking. So the movie is playing in reverse now. So now you see how the octopus is becoming the bush. So you see the color, the texture, the scales. And at some point, you really cannot make out that it's an octopus. How is it controlling these patterns? I mean, one is to understand how patterns form and write down the physics of that, maybe some instabilities, maybe this and that. The next thing is to be able to control that. And do you know how much time it took to go from that pattern to this pattern? Okay, you see this? Time scale, 2 seconds. So this is an object which is a couple of tens of, maybe a few 100 centimeters, let's say, or a few tens of centimeters. And it's able to control this pattern in a matter of 2 seconds. So you should make some estimates. Is this diffusion driven? Is this advection driven? Can you actually flow material so fast from one region to the other? How is it that it even does this? So, I mean, one of the most fundamental things, camouflage. Why is it that these things can actually control them so far? I mean, you can ask a why question like, okay, it's useful, it's prey, predator, it wants to hide. How is it? So one doesn't know. I mean, one just is exploring these kinds of patterns. And so the current idea seems to be that they are controlled directly by neurons. So therefore, that brings in a whole different biology and connects it with pattern formation. We are not going to touch that at all. We have no idea of how these things work. I'll do some very mundane, boring things for the lecture. So my idea was to show you that the kind of patterns that are there in the natural world, in the living world, are bewildering, and the way organisms seems to use them, to control them, is shocking. Okay, so how do we do this then? By the way, for this video, I would strongly recommend there's one more video called Kinks of Camouflage. It's a PBS documentary. It's available on YouTube. Take a look at that. Cephalopods, which is the name for these kinds of creatures. Can you give me host permission? Okay, that's you. Cephalopods, which are basically cuttlefish and octopus and a bunch of other creatures, they're masters at camouflage. And how they actually control such patterns, how they use those patterns to stun prey, to escape, to hide. That video is really gorgeous. Okay, so how do we model these patterns? I mean, should we start from maybe quantum mechanics? Or write down a more very detailed description of the game. So it turns out that you can do that, but we'll soon realize that the problem becomes insurmountably complex. So therefore, you can't do anything about it. In principle is one thing, but in practice, you can basically say nothing about it. So in fact, there is a nice paper by David Pines and Robert Laughlin in PNAS. I think I had the slide and I missed it here. The title of the paper is Theory of Everything. So I very strongly recommend you to read that. And there's only 2 equations there. There seems to be, maybe I don't remember if I have it. Just check. Yeah, I do have it. Okay, cool. Okay. Okay, so how do we build a theory of pattern formation? So one is to appeal to some very microscopic laws like laws of quantum mechanics or whatever. But then the thing, the description becomes incredibly complex. So therefore you really can't say anything much about it. So then, okay, it's just a matter of in details or no, the idea is different. I mean, the whole point of this kind of an approach was something laid out by Anderson. I mean, if you haven't read this, must and should, everybody in science. One is to think about a reductionist approach. Let's go down to the bottom levels. Let's look at the most fundamental things. And then say, in principle, I'm done. Anderson argues that's wrong. I mean, the point being that because you know everything at the bottom level, does not mean you can reconstruct things at the higher level. So at the higher level, there's a new set of laws that come in there. These are emergent laws. And in particular, at each level, there are new properties that come up, which you cannot even predict, even in principle at the bottom level. And so therefore, the study of these kinds of objects, basically statistical physics, is as fundamental as anything else, right? So it's important to remember that. And so the idea is really of emergence. How do you actually get these kinds of emergent structures? So this paper, the title is indeed The Theory of Everything. And if you look at the paper, they just say, hey, for almost everyday life, you just include light, electromagnetism. This is the theory of everything. There is everything that is there. But then you realize, how do you predict the octopus patterns from here? That's quite difficult, right? So they go on to give a similar approach to what Anderson says. The idea is that there are emergent rules that you really need to bother about. And in practice, that because you have a fundamental description does not mean you have an emergent description there. So emergence typically requires the following input. This is something crucial. You cannot take a Hamiltonian and reconstruct everything from start. Emergence really means you have to interface with experiments. There are certain points at which you want crucial experimental input. Some number is large, you can neglect some effect. Some number is small, so therefore you have to worry about that. So this comes from really looking at data, experimental data. And that is very important to take care. So that's the approach that I'm going to take about to build pattern formation. Okay. So I'll dive into a little bit of the details from now on. Please do stop me if something doesn't make sense. So the approach that one is trying to build to understand even pattern formation is what is called classically the, how is equation 1 and 2, the theory of everything in the, so I would strongly recommend that you read that paper. They will try to explain that everything in the everyday world can be explained with that set of equations. Okay, any other questions, comments? All right, the theory of everything is fine. So the approach that one tries to do is what's called a hydrodynamic approach. So the idea is that, hey, I know that if you take a bunch of objects and put them together, and if you don't slosh them, squish them, move them, heat them, don't do anything to them, they reach thermodynamic equilibrium, Stat 101. And so there are static variables, thermodynamic variables like pressure, temperature, chemical potential, blah, blah, blah. And these are things which are independent of space and time. So there is a pressure, a temperature, a chemical potential. So if you try to take such a macroscopic system and perturb it slightly, not very far, but slightly from equilibrium, surely there's going to be a spatiotemporal dependence to these thermodynamic fields. And so therefore you would like to understand how these thermodynamic fields, which are now space-time dependent, will drive fluxes which will tend to restore equilibrium. And the theory for doing that is basically hydrodynamics. And the classical example of such a theory was fluid dynamics. Hydro does not mean water. This is generalized hydrodynamics. The theory for liquids was probably the first example of this kind of approach. So the idea being that you can think about a local thermodynamic equilibrium in the sense that every tiny pocket can be assumed to be locally in thermodynamic equilibrium. Locally, it has reached some entropy maximization, but the neighboring packet is not. And so therefore, there is a temperature difference. Maybe there's a pressure difference. Maybe there's a chemical potential difference. So therefore, there will be currents. How does one describe such currents? So these perturbed fields will generate thermodynamic forces. And the forces are such that the fluxes that result from them will tend to equilibrate the system. So classical example that we all study, right? Temperature difference, there's a current which will try to equalize the temperature, pressure difference, chemical potential difference. So left to itself, the system will of course reach equilibrium after a long period of time. This is different from non-equilibrium steady states, in particular the kind of patterns that I'm talking about. So these are systems where you constantly supply energy into the system. So therefore, you do not let the system reach thermodynamic equilibrium. So in a sense, you do reach a steady state because things don't seem to depend nominally on time. But it's not because it's reached some thermodynamic equilibrium. It's because it's reached some sort of a non-equilibrium steady state where there is a constant consumption and dissipation of energy. And yet the probability distribution to find these macroscopic states is time independent. The point to note is this is being cut off here. We don't have a non-equilibrium statistical mechanics in general as much as we have an equilibrium statistical mechanics. We have loads of tools and theories and rule of thumbs, and these things do work most of the time. But a generic theory like of equilibrium is something which we still lack. Okay. How does one build a hydrodynamic theory? Well, here's the idea. Just something again. Can we get this off, if you don't mind, IT people? All the references will go off otherwise. Don't press the N. How will that help? No, there usually is something like hide this, but it's not here. It's not here. Is that you? This is my laptop. Okay, that's my cursor. This is not my cursor. Okay, anyway, so once the IT people fix it. So the general idea is something which is the microscopic fluxes are reversible in nature at the local thermodynamic length scales. Yes, if the system is at thermodynamic equilibrium or close to thermodynamic equilibrium, but if you have a system which is fluxed with energy and is driven out of equilibrium, then you do not have time reversal invariance. Equilibrium is time reversal invariance, non-equilibrium, you're broken time reversal invariance. So I'll just come to that in a minute. So the idea is basically the following, that you have a many-body system, a glass of water or maybe a pneumatic liquid. And so you disturb it away from equilibrium. So maybe you slosh it, maybe you heat it, whatever, and leave it there. And so it'll relax. So most of the degrees will relax very quickly. But there'll be a few of them, which are more like the collective degrees of freedom. And these things take a long time to decay, long compared to the kinetic relaxation timescales. Think of the typical molecular relaxation timescales. And so these are what are called the hydrodynamic degrees of freedom. These are the long-lived modes. And the relaxation time of these modes will diverge as the wavelength of the perturbation goes to infinity. So I'll work out these things a little bit in detail, maybe tomorrow or the day after tomorrow. So some examples, for instance, if you have a conserved quantity like mass or momentum, the densities of those conserved quantities are hydrodynamic variables. If you have a phase transition, close to a phase transition, or you have an order parameter that describes a continuously broken symmetry, for example, the polar order parameter, the densities of such variables are very good hydrodynamic variables. So here's an example. So you things like scalar fields, like densities or order parameter fields. Okay, there's another thing cut off here. It's called polar. Or you could have things which are pneumatic orientation fields, flow fields, or polar order parameters. So these are things which are very good examples of hydrodynamic theory, hydrodynamic modes. Now, the thing with biology, and this is something I will work on a little bit in the next few classes, is that in addition to these classical fields of densities, orientation fields, and so on, there are also chemical reactions. And this is something that you cannot get rid of. And typically, biological patterns are really what are called mechanochemical patterns. They inseparably couple mechanics and chemistry. So for those of you who got scared of chemistry with looking at those benzene rings, I also got scared, so I'm not going to touch that kind of a chemistry. For me, chemistry would basically mean some kind of polynomial terms that you would put in the densities of these quantities. And so that's the level that one would like to look at the pattern formation level. And so the coupling between these kinds of things leads to new set of theories. Classical example of hydrodynamic theory, diffusion, right? So there's the density field, which you can write from fixed laws. And again, everything is cut off. Can we fix this, if you don't mind? Nobody there.
Vijay, I think there's one question is, are the microscopic fluxes
I answered
reverse?
I answered that. Yeah, I saw that. There's nobody there. Okay, I hope you can assume this is not a minus sign, this is an equal to sign. This is derivative of temporal derivative of C.
I sent a message.
That's the diffusion equation. So classic example. So I presume this is familiar to everybody. If it's not, please let me know. Okay. So classic idea of diffusion, right? So that really comes from, as we all know, from the random walk picture. So if you have a particle doing a random walk, you can look at its mean square displacements. And so if you look at the a sort of a density field, which you define as a bunch of Dirac delta mass points, you can actually construct the equation for the density field, and that turns out to be the diffusion equation. So you have a bunch of particles doing a random walk. Okay. All right. Now that's probably the simplest picture that one can think about. So things moving around by simple random walk, simple model. This is itself quite good for establishing many patterns in biology. Turns out that one of the first examples of how diffusion is used to generate patterns was something developed by Francis Crick. So he basically said that think of the embryo, the fly embryo that I showed you, and think about having a source of some molecules here, so C left, and another reservoir of molecules on the right. And the molecules are just moving around by diffusion, simplest example, right? So can you actually say measure the concentration fields and check if that's diffusion? And so obviously you all know that in the steady state you get a straight line profile of the concentration field. Turns out that people did do that, and it's from the past 2-3 decades that people have been actually able to measure these concentration fields quite well. So you remember the fly wing that I told you about? The front and the back of the wing get patterned by a certain molecule being sourced at the center. Now, why is it that molecule is sourced? Okay, that goes into a bit of biology. So take that as granted for now, that there is a source of this molecule at a particular point in one dimension. The molecule just moves around by diffusion, and it's degraded at a certain rate. The molecule will get degraded by interacting with other chemicals. Surely you can set up this equation, solve it by TC2. And you can actually measure the concentration fields of these molecules. Turns out that this is a pretty good description quantitatively. Why is it useful? Well, it turns out that this can be used to build certain other patterns. So here's one way in which the stripe patterns that I showed you in the fly are being thought about. So let's say you have some molecule which is being sourced at the left corner, that's molecule A, and that sets up some exponential profile. Okay, so that's one non-autonomous input. So you put more of A on this side and let it go. So diffusion and degradation will set up. But now you can have a bunch of chemicals where, let's say, A makes more of B and C, but A reduces the amount of D, so that A, B, C, D sets up some other. So there's something else between C and D, and there's a complex chemical reaction network. At the end of the day, you'll end up with very nice periodic patterns. So there was one external input, the rest of the thing was self-organized. Well, is this really there? Turns out yes. So these stripe patterns that I showed you, you can actually mark each one of them with some different colors, fluorescence techniques in biology, and turns out that each one of these has a name. You can actually measure the concentration fields. And so this reaction network that you thought about as composed of simply diffusion, degradation, and one source at the end is a pretty good descriptor of the stripe formation patterns. It's an example of what's called a controlled pattern formation, because the boundary that you put in really basically controlled everything. The rest of the system was self-organized. Another example is something where you don't have anything which is non-autonomous, Turing patterns. I'll work out this in gory detail in the next 2 lectures. So I just want to give you a feel for what is going to come up. You have a system of 2 chemicals, at least 2, and you have them diffusing in some background, say in 1 dimension, or could also be in higher dimension. And there are chemical reactions between them. So U and V represent the concentration fields of the 2 species there. If you actually look at how the concentration fields change, that this is all that is to it. They change by both diffusion and reaction. So here's one example of some model called the Guérin-Maynard model, some choice of chemical reactions, which seems to be useful. Turing showed the the same Turing of the Turing lectures Turing. In addition to computer science, he also did fundamental biology. He showed that if you have a bunch of chemicals like that, at least 2 of them, and you satisfy certain criteria among the diffusion constants and the chemistry in the sense of not particular molecules, particular reactions, but basically something like, is one molecule something that makes more of itself? Maybe there's one molecule, the inhibitor, which reduces the activator. We'll work out the details as we go along. He showed that if you have this, this is actually enough to give you very nice periodic patterns, what are called Turing patterns. And so it's a bit of a puzzling thing when you think about it. Diffusion, you normally think it's going to homogenize stuff. And here I'm saying something where I have a combination of diffusion and completely local chemical interactions. So for example, think about this model. Oops, sorry. This model for reactions. These are completely local. The U and V there interact with the U and V there. No spatiotemporal long-range couplings. And this theory is saying that this is enough to give you non-trivial spatial patterns. It's a bit puzzling. So diffusion, I thought, is always going to homogenize stuff. Now we are saying, no. Diffusion can actually drive a non-trivial spatial pattern. How do we understand that? So these criteria and all, I'll work out when we do that next time. So I told this to Abhishek, and I'll also emphasize this. Most of pattern formation is well appreciated if you actually do some numerical calculations. Otherwise, it becomes very difficult. You can do a linear stability analysis, maybe some weakly nonlinear analysis. Beyond that, it really becomes hard. So my hope is that at least in the tutorial, some of you will be interested. I can give you Python codes to actually develop simple codes to solve these equations. Not at all difficult, few tens of lines, and you'll be able to solve them in some higher level language, say Python. And my hope is that by the end of the day, you'll actually be able to go from a homogeneous pattern, like you started out with the U and V concentration fields in one dimension. And once you satisfy some Turing criteria, the homogeneous state becomes unstable, and you will spontaneously get these periodic patterns. Hopefully, you will be able to do this by the end of the lectures. So these are self-organized patterns in the sense that you don't need external non-autonomous input from the surroundings. And these occur due to instabilities of the homogeneous state. They spontaneously break the translational symmetry, translational and rotational symmetry of the uniform state. So you can get like waves and spots and stripes and labyrinths and blah, blah, blah. There's a trouble there. And this is probably the reason why Turing patterns were not liked by biologists for a very long time. These are uncontrolled because they occur due to an instability. So in the sense that they're very sensitive to initial conditions, how many stripes will come up, where will they come up? These are all things which are a bit dicey. So here's, for example, a simulation of this, some set of reactions, Gyra-Maynard, something that you should do. And in one set, you get some space-time plot. The color indicates the concentration field. And you get some nice stripe patterns after a certain point. You redo the same simulation, same set of differential equations, same set of parameters, but a slightly different set of random initial conditions. And you get a slightly different set of stripes. So points like, where do the stripes come? When do the stripes come? These are uncontrolled. Now that's troubling for biology. So most people try to model, many people try to model all periodic patterns in biology as Turing patterns. I mean, there's a whole industry running around in the 80s and 90s. It's only from the past decade or so that we've got quantitative confirmation that biology actually uses the Turing mechanism to really build patterns quantitatively. Qualitatively, we've had that from several times. Perhaps the first one was the work by Kondo, who showed about fish stripe patterns. The more recent one is digitation patterns. In
this earlier equation, you are also putting things from outside.
No.
No, the previous one, because there is this term A, right?
Oh, there's a flux. There's a non-equilibrium flux that you maintain.
You put it from outside.
Yes.
Right? So it's not like a self-placing, but you're also putting things from outside.
So this is a system which is driven out of equilibrium. So there must be a source of energy somewhere. That is all there is to it. So I was trying to contrast it with the case where you control something at the boundary by hand. That is not the way this happens in the bulk as well. So classical example is of digit pattern formation, digit formation. So 5 digits that in your hand is classically been thought of as Turing patterns. And very recent work seems to show that this is indeed the case. in the sense that you can actually measure the concentration fields of the chemicals, measure their interactions, show that they satisfy this criteria, and there is also a way of controlling those patterns. Okay, so that's Turing patterns. Basically, how can we say it is uncontrolled as it is greatly being controlled by the initial conditions? Yeah, so the point being that these are nonlinear differential equations. And so you can typically have multiple possible states at the same time for the same set of parameters. And that is something that I will actually use it later on. And so therefore, where you put the initial condition very precisely really depends, really dictates where the pattern goes to. Now, in a real system, that is bad because there will always be fluctuations. There will always be noisy states coming up every now and then. And so if you are so sensitive to the initial conditions, you're in trouble because I want to get 5 fingers and at a particular point of time. And that shouldn't depend on how much noise there was. So therefore, that's why basically these are things that come from instabilities, and that word itself should tell you that instabilities are hard to control. Is that fine? People can open and talk also, right? Or they have to almost have to type. Might be easier if they open up and talk.
Open, right?
So people can also open up and talk rather than type. Easier. Okay. These are reaction diffusion patterns, the 2 patterns that I showed you. These are basically things which have, say, one molecule, some boundary control term, and you get some pattern. Maybe 2 of them, one here, you can get some nice kinds of pattern. The other one are the Turing patterns, which are self-organized. And these are things which spontaneously generate the patterns that you see. There's a catch. And this is something that people have thought about for a very long time. There's no mechanics, there's no flows, there's no forces. And it's very evident that if you think about any kind of a morphogenetic pattern, the developmental patterns that I showed you about, patterns in shape, for instance. How can that be that these are things which are occurring without mechanics? If I give you a piece of clay and ask you to mold it in the shape of a fish, you have to push, pull, squish, slosh. That's a stress tensor. That's a mechanical force. How is that not appearing? Turns out that they are there. We didn't know the components which actually make up this mechanical force generators. And recent work has really cast a very good description for these kinds of problems. Okay, yeah. Cell division, like I said, is one of the most fundamental things which distinguish living things from non-living things. So this is all the biology that you need to know. Just remember there's something called DNA which stores stuff. And then there are things called proteins which actually do all the work. So most of the time, if I show you something which is brightly lit up, it's a protein, very likely. So cell division that happens here is really orchestrated by these proteins. These long fibrous things are also proteins. So just look at that movie again and again. You see a bunch of DNA molecules which are condensed here and they're being pulled apart, right? Shouldn't there be a force to pull things apart? And indeed, the idea is that the cell has these structures called the cytoskeleton, which is like a skeleton which provides the scaffolding for the cell. And active processes in these cytoskeletal materials actually drive force, any mechanical phenomena inside living systems. So there's a very nice set of theories that have been developed. These are what are called hydrodynamic theories for active materials. And again, I don't expect you to get a lot. I will derive some of them, not all of them. I'll just motivate some of them. And so you can look at pattern formation in these kinds of things. So for example, the structure which is just beneath the cell, called the actomyosin cortex, turns out to be something which is very crucial in cell division or cell polarity. And patterns in these materials actually drive many morphogenetic processes. Okay. So let me skip some of them. So here's, for example, a model for a pattern formation in a cortex. So this is something I will work out in detail again. So let's say you have a single molecule A. That molecule is moving around by diffusion, simple diffusion. But there's also a flow of material. What material and all will come to that. But this molecule is special in that it generates mechanical forces, mechanical stresses. So via some fluid dynamics, there is a fluid dynamical equation for the flow field, and the flow field couples to the transport. So this provides a sort of closed set of equations, though it's out of equilibrium. And you can show that if you work out this system, this has a spontaneous pattern forming instability. In fact, if you put in a simple turnover, it gives you very nice periodic patterns. So this is something I want to work out maybe by the last class. Okay, so that was the basic introduction that I wanted to give you a panorama of the kind of things that I want to work out. And what else do I have? That's all. And the general picture that one is trying to build is that, hey, if you have cells and proteins and genes and networks, okay, there are all these structures. So there's a statmechy way of thinking about phenomena. So that is, if you give me a bunch of things, put them together, you know the interaction. There's a formalism called statmech or thermodynamics, which you have mastered. We can create a metro or an aeroplane. For life, there's genes and proteins. There's no interactions about them. There's lots of them. And there's giraffes. I don't know how to go from here to here. So that's a big question mark, which is widely gaping open. That's the kind of approach that one would like to take. Okay, so I'll now switch to the board and start working out some simple models for pattern formation. So my basic idea is to first go through a dynamical systems perspective, think about fixed points, steady state, stability, and lay out this for today. And from tomorrow, start with partial differential equations with some simple models for pattern formation, maybe in living or non-living systems, and then go to reaction diffusion patterns, work out Turing. Hopefully, you permitting, I'll go a little bit faster on hydrodynamics and develop active matter. And the concluding 1 or 1.5 lecture will be on pattern formation in active matter. And the last half an hour or so, I'll give you some broad feeling of the kind of problems which are open. We don't have any idea about them. Questions, comments on the stuff so far before I go into the details? Yes, sir.
So the UV field equations.
Louder, please.
So the UV, is it?
Yeah, go ahead.
So the UV field equations you showed.
Yes.
So I mean, those look. I mean, you can look at the telegrapher's equation with some extra terms in that way, right? I mean, it's just like a single particle RTP. Plus P, I mean, the probability with right.
So the reactions are nonlinear terms here.
Yeah, maybe you put something, say that the
reaction nonlinear terms are crucial here.
Okay, so that condition you showed that I mean that there's some more things to when this, like the pattern formation.
I'll work out that condition. I'll derive that condition. So I mean the nonlinearity is important. I mean if you have completely linear PDEs, basically some eigenvalue problem, right? At the end of the day, and the boundary conditions will essentially determine the structure that you have. Anything else? Okay. People online, no questions, comments? Comment, suggestion, please indicate a little more. Yeah, sure. I'll put in all my notes and I'll put up on the website. All right. Okay. So the picture that I'm trying to develop for pattern formation is one of dynamical systems theory. And so it's good that we go through a couple of calculations for dynamical systems theory. So we don't need the board for the rest of the class. We can push it up and switch off the presentation as well. Is this visible, everybody? So I want to look at and the picture that I'm going to take is one of a dynamical systems perspective, which means I'm going to look at differential equations, ordinary and partial differential equations, and we'll develop the tools required for trying to understand pattern formation in these systems. So what will I neglect? I'm not going to worry about fluctuations. So these could be intrinsic noise in the chemistry itself. Perhaps Supriya will cover some aspects of that. These are because the number of molecules is very small, so there are fluctuations there. There are also hydrodynamic fluctuations that you need to put in typically into the PDEs. I'm going to neglect those things. Okay, so what you have then is a system of differential equations ordinary or PDEs, and I want to look at non-trivial solutions to these equations. And so to develop the perspective, so what I'll do is to start with a simple ODE dynamical system and develop the idea of fixed points. So at any point, if this is obviously clear to everybody, just let me know and I can speed up and do other things. Oh, these are both the same thing. So there are two of them. Yes, you can. Because you can have any non-spatial system, well-mixed system, but have small number of molecules, and you can still have these intrinsic noise. So these are things that come because the chemistry itself is stochastic. There'll be fluctuations there, and think of you were there in my class, you should know this. We had worked out the Poisson, you're there, right? Yes, we did. So if you have even the simplest reaction where something is being produced and something is dying off, you can work out the probability distribution to find n number of molecules of A at some point of time. And it's only in the limit when the number of molecules is very, very large that you can talk about a number of molecules. Otherwise, there's always a fluctuation. This would typically come about when you have spatiotemporal degrees of freedom. Right? So if you have a coarse-grained description where you have some dissipations and fluctuations, they can be related by fluctuation dissipation. Okay, so very abstractly, I have a field U, and it could be a single field, could be multiple fields. And what I have is an evolution equation for U. And there is some equation that governs this. So in principle, it could be f of ut, but I'm going to neglect that for now. And I'm just going to look at f of u, an autonomous dynamical system. So the first notion that you think about is the idea that there are fixed points. So these are basically points where f of u star is 0. So that means the system, once it reaches u star, does not evolve in time. Okay, so that's a fixed point. Okay, you can easily convince yourself. For example, here's an example. If I give you d by dt of x is sine x, please figure out, for example, what are the fixed points for this system. Any n pi, x equal to n pi is a fixed point, right? Okay, so x equal to n pi is a fixed point. So the first thing to notice is that n is an integer. There are multiple fixed points. And so now if you say, if you start the system somewhere and allow it to evolve under this dynamics, which fixed point will it go to? How will I know which fixed point it goes to? So I'll sketch F. So you said there are these three fixed points. Now, which fixed point will it go to? Depends on where I start from. Does that make sense? Yeah? Okay. So let's say I start from somewhere here. So can you tell me which fixed point does it go to? Go to 0 or does it go to pi? Is the question clear? Or is it so difficult that you can't answer?
Pi.
Pi? Okay. I don't know how, but how did you figure out that? Okay. The velocity is positive. Huh. Okay. Okay, so what intuitively you're trying to get is, A, there's fixed points. So this is finding the fixed points. The next idea is the stability of the fixed points. So that means that if I have a fixed point, if I have reached the system at that fixed point, is it stable? Stable to what? Stable to small fluctuations. Suppose I go around 0 and I perturb the system slightly. Does it come back to 0 or does it go away from 0 and go to some other fixed point? So that is basically a question of the stability of the fixed point. So there's fixed points and the stability of the fixed points. And that's something you need to really work out. Okay, how would one find out the stability of the fixed points? So the stability requires the following idea. So you perturb the system around the fixed point. So that is, you do a perturbation of the following form. So this is a perturbation. And you ask, if I do this perturbation, does delta U grow in time or decay in time? In other words, does the system come back to U star or does it go away from U star? So if the first one happens, you say the fixed point is an unstable fixed point. The second one happens, you say this fixed point is a stable fixed point. So intuitively, what you can think about is a particle going around in some potential like that, intuitively. And if you have a particle at the height of the potential there, surely any displacement, either way, is going to take it away from that point. Even though at that point, particularly, the force is 0. On the other hand, if you look at any of the valleys, any small fluctuation will bring the system back to the same fixed point. So therefore, these valleys are basically like stable fixed points, and these mountains are like unstable fixed points. Is that fine? So I'll develop a formal way of looking at that. Now, one is to understand the stability of the fixed points. And typically, surely we want to look at some non-trivial solutions. At the end of the day, this will basically become some reaction-diffusion equation, maybe some polarity field, some flow field, and so on and so forth. And there'll be a bunch of parameters. So what you would like to find out is, given a set of parameters, which fixed points are stable, which fixed points are unstable. So you would want to follow the stability of the fixed points. So here's one example, classical example, and I want you to work out the fixed points and also their stability. So I'll give you an equation which is dx by dt is r times x minus x cubed. So that's my f of x. Sometimes I will switch x and u, but think of u as a general. So what are the fixed points here? So there is 0. Surely that always satisfies that. Anything else? Huh? Plus or minus square root of r. Okay. I want to restrict my x to be a real number. And so therefore, what about the second fixed point? Does it even exist always? It depends on the parameter r, right? So if the parameter r is non-negative, surely this fixed point exists. And so there are really 3 fixed points, 0, plus root r, and minus root r. But if r is negative, then this fixed point doesn't even exist. So for r less than 0, let's go one by one. The second fixed points, second and third ones do not exist. It's only x equal to 0. I would like to find out what happens is x star equal to 0 a stable fixed point. What do you think?
Yes.
How did you get that?
because the derivative of Rx minus XQ about X star is equal to negative.
Yeah, so that's basically the idea that you would want to do. So what you want to do is to do a perturbation, which is around the fixed point, and then ask how the perturbation grows. Now, surely if you do this perturbation, you put it into the equation, well, the equation is nonlinear, you still get a nonlinear equation. So you won't be able to do much. So what you say, I'm going to do a small perturbation. So which means I can do a truncation in powers of delta x. And so if I retain things to lowest order in delta x, then I can figure out what happens to that perturbation. So let's do that. And can you please tell me what is the d delta x by dt? Please do the algebra and let me know quickly. So in general, this will basically turn out to be the following. So you will evaluate df by dx at x equal to x star, and you will get it to be delta x plus higher order terms. Can you please see if this is true? Because remember, x star is the solution of f of x. So if you do a Taylor expansion around x star, f of x star is 0 by definition. Aha. So therefore, it's really the sign of df by dx that really matters. And so for this particular example, can you please work out what is df by dx?
R into delta x.
R into delta x. Okay? So which immediately implies that delta x is. So therefore, if R is negative, it's guaranteed that x star equal to 0 is a stable fixed point. Now, for R less than 0, less than or equal to 0, that's all there is to it. I mean, there are no other fixed points. So when R is positive, please work out the similar thing. Same question is x star equal to 0 stable now? We don't have to rework. We already have the answer. You should be able to tell me quickly. Is that stable? Unstable?
Unstable.
It's unstable. Because you just said that this is an exponentially growing increase in the perturbation. And if R is positive, very soon x star, the system will leave x star equal to 0. So no, this is not stable. All right. What about the other ones? So you have the other two fixed points. Can you please work out their stability and tell me? So I do a similar perturbation. And I want you to tell me how this perturbation grows. Something, something, something times delta x. So what is that something?
Minus R into delta x.
Minus R into delta x. And is this guaranteed to give you stability? R positive? Yeah. So this will tell you that this will always decay. So delta x, the perturbation, exponentially decays. So which means the fixed points, x star, which is plus minus root r, are stable. So what happened here? I mean, okay, so let's draw what's called a bifurcation diagram. So where I'm plotting the fixed points and the control parameter r, x star, on the two axes. So for r less than 0, x star equal to 0 is the only fixed point, and that is stable. For r greater than 0, x star equal to 0 is unstable. But at the critical point r equal to 0, these two fixed points are identical, and the plus minus root r fixed points bifurcate. what happens to the x star equal to 0 fixed point? Is it still there or it's disappeared? For R positive,
It has disappeared.
it's disappeared. So for R positive or negative, is x star equal to 0 always a solution or not a solution? It's always a solution. This may or may not be a solution in the real domain if R is positive or negative. So this will always exist, but what's happened is it's lost its stability. So that means what I do is to indicate that one by a dashed line. More like root x. Okay, so there's the root r fixed point and minus root r fixed point and the 0 fixed point. So this is a classical example where you have a bifurcation in the dynamics of the system where a single fixed point bifurcated into two, the x star equal to 0 fixed point lost its stability. And what you got was two other non-zero fixed points plus or minus root r. So in a sense, that's the crux of pattern formation the con will do. So what we'll do is to make things more complicated. We'll make it PDEs and many variables and tensors and vectors. At the end of the day, what we'll do is to figure out what are the control parameters in the pattern forming system. I'm going to tune the control parameters. Some states lose their stability. Some other states gain stability. They may appear and gain stability. So this could be the homogeneous unpatterned state. And this could be the pattern states. Okay. So what we did here was basically the idea of what's called linear stability analysis. So I'll set this up in general terms and illustrate this with a bunch of examples. And this will be our first go-to tool when we want to discuss pattern formation in either reaction diffusion systems for Turing or active mechanochemical patterns.
Vijay, there's a question from Fazil Najib. Are we controlling R?
Are we controlling R? Yes. R is, well, think of it like a temperature, pressure, chemical potential, some parameter, some external control parameter that you're controlling. Or you could think of this some sort of a phase diagram that's appearing in these control parameters. So you change R, and as you change R, some states disappear, some states lose their stability, new states appear. So far, so good? Yeah. 1, 2.
Same question. Yeah. What happens when R is equal to 0?
R equal to 0, the fixed points are coinciding, no?
To the stability.
To the stability in the sense?
Like
which fixed point?
X equal to 0 will be the fixed point.
Yeah.
Then
it's neutrally stable. It doesn't grow or decay.
Okay.
So therefore, at the linear level, it doesn't help. You have to go to one more higher order. Okay. Just like Taylor series, first derivative vanishes. You have to go to higher order. There's one more. Now, for one-dimensional systems, basically one variable, I mean, at the end of the day, you can really think of this as some sort of a potential. And you can really appeal to this picture that I'm in the minimum of the potential or the maximum of the potential. And so therefore ask for whether it's stable or unstable. Well, not so if you have more than one variable, right? Because you know that even if you have a simple ODE system of two variables, you cannot in general write this as the derivative of some potential energy. And some conditions have to be satisfied on the force function and so on. So if that's the case, then you cannot in general think of a potential energy. So one would like to develop a more general mechanism to do this. I should stop at 12:30 exactly. Okay, I'll take a 5 minutes or so extra there. All right. Okay. Now, what I did was to write down a differential equation very abstractly, and I mean, where's the physics there? I mean, is it Newton's laws? Is it? So how about the following? So let's say I actually have the following system. I would like you to work out these details. So let's say I have a particle in a potential of this kind, which is like r x squared by 2 minus or plus something. What did I didn't put anything? So x4 by 4. With a minus sign here. Just do Newton's laws. I mean, you do x dot. So now we have two variables, x dot equal to v and v dot equal to du dx. So it's not at all difficult to figure out the dynamics of the system. And it's quite easy to do the calculation again. I urge you to go through this system and you will be able to show that the same picture comes up. But here you'll need to do something more. Now we have two variables. You'll have to develop a general way of thinking about the system in terms of matrices and their eigenvalues. How will you do that? Well, here's the idea. So what I have in mind is a system which is under the influence of this potential, but also has a damping. You can put some gamma and non-dimensionalized things appropriately. So I'll set units such that this unit, this system is 1. So what are the fixed points? So again, the fixed points are at x equal to 0, one fixed point, but this time also with v equal to 0. And the other two fixed points, x star equal to plus minus root r, but again with v equal to 0. So do the perturbations again. So you can do x equal to x star plus delta x and v equal to v star, which is 0 plus delta v. And if you put this in, do the Taylor expansion, the claim is the following, that you will end up with a system which looks like this. Some matrix times delta x. Okay, please do this exercise. It's quite easy, trivial to do this. And now the matrix A, which is basically the linear stability matrix, essentially the Jacobian. And the properties of the linear stability matrix will dictate the stability. So I'll probably do this the next time. But what you can show, which we'll do next time, is it's really the eigenvalues of A that matter. So if you have an n by n system, here 2 by 2, n eigenvalues, look at all the eigenvalues. Some of them could be complex, some of them are real. Look at the real part of all the eigenvalues. If the real part of the eigenvalues, you sort them. If the eigenvalue with the largest real part, eigenvalue with the largest real, has its value negative, then you can show that this system is completely stable for that fixed point. Even if there is one eigenvalue whose real part is positive, that fixed point is unstable. And so you can go away from that fixed point. So maybe next time I'll develop this in more detail and set up a general formalism to show you that this fixed point is unstable. Yeah.
Vijay, there are, I think, a couple of questions.
Yeah.
I mean, one is here we linearize a nonlinear system. Can the higher order terms affect the stability that significantly, or the nature of the dynamics around fixed point?
The higher order terms affect.
Can the higher order terms affect the stability that significantly?
Yeah. So if it turns out that the derivative is non-zero, then to small perturbations, this is a good enough approximation, and this will be the dominant contribution. The higher order terms will only give you insignificant contribution. If the first derivative is 0, then those are the leading order contributions. So you have to worry about it.
Vijay, in the same spirit, I had the question, like if this if it's not a small perturbation, then how, like this.
If the perturbation is not small, what happens? Then there's very little one can do.
That one gets from linear stability.
I mean, so if it's linearly unstable, then it's definitely unstable. If it's linearly stable, doesn't mean it's stable. It could be non-linearly unstable. So if the perturbation is large enough that it can go beyond that, go beyond some hill, of course you can get determinant. So you can't predict that directly from here. There are a bunch of other things.
Can you repeat the point about the eigenvalues of the stability mechanism?
I'll work out that in detail tomorrow. So I'll show you the eigenspectra and talk about the linear independence of the eigenvalues.
Yeah, that's
okay. Okay.
This Prashant's question, you always have a domain of stability, actually, right? I mean, like, if you go beyond that domain, that you will go to a defined fixed point.
That's perfectly correct. Yeah. So where is that sin x thing that we had? So there'll be some domain of stability here, basin of attraction, right? So it's within that you will have a stability. And if you're doing linear stability, you're only doing some perturbations around the fixed point for very small values, right? If you surely do a large perturbation, linear stability can't tell you anything. It's basically an approximation of that high dimensional function by a straight line. How good is that approximation a straight line? Well, as long as you're close to the fixed point, not far from it. Okay, so my hope was that in the first lecture, we'll set up this basic thing. Tomorrow, I'll do a detailed calculation of the linear stability. And from there, we'll go to the linear stability for PDEs as well, where I'll have to look at Fourier modes. Okay, some feedback would be useful. Is this too fast? Is this too slow? Is this everything is too evident? Everything is boring? Another possible one is go to hell. I don't care what you say.
Amazing.
Something would be useful. So I can speed up, slow down. I can go at the same pace. I can. So the default answer is the last one that I said. So.
Excuse me.
Yeah, go ahead.
There was this one point I could not understand where you wrote x dot equals v and v dot equals minus del u by del x. So is it v or is it gamma? I couldn't understand.
Is it v or?
Yeah.
Repeat that.
So in the first equation, you have written x dot equals to v.
Yes.
Right. I can sense that it's if it is position, then that's velocity.
Yes.
And the second term, you're writing that v dot is equals to minus del u by del x. So that needs to be 4. So
yes.
I mean, is it the same thing?
Yes. Newton's laws.
I see. Okay.
So this is some frictional damping. I mean, you could have some small gamma or b. I've just said I've non-dimensionalized things and put that to 1. Is that fine?
Yeah, no, it's fine. Thanks.
Huh? So I don't know. Should we go to the tutorial today, or how should this work out? Yeah, we can work out. No, I don't want to do the eigenvalue thing. I'll do tomorrow, but I can work out some problems as well. Some homework problems. Yeah, do this stability and show me. That's one thing.
Excuse me, I have one doubt. How is a fixed point is square root of r in this case?
So I'm trying to solve rx minus x cubed equal to 0. So therefore, there's one obvious solution, which is x equal to 0. And the other solution is x equal to plus minus root r.
But if we solve the first equation, which is x dot equals to minus rx square by 2 plus x square by 4, in that case, we will get like square root of 2r, right?
Okay. I might have screwed up something here. I think it's fine, as far as I can see. It's fine, no?
Oh, okay. I was confused.
Do the derivative of this with a minus sign and you'll get back the same.
Okay.
Yeah. Yeah. The second determinant? Yes. Yes. There'll be a 2 here. Okay, maybe, I don't know. All I cared about is there's a minus sign for sure. Then ensure stability. Again, work out these things. Okay, fine. Yeah.
Regarding that, from the introduction of
yeah.
So you mentioned for the broken detailed wells,
yes,
the consumption has to be at the small length scales as well as the distribution. Is it both of them?
Repeat that.
The consumption
yes
of the energy must have to be at the small length scales as well as the distribution. Is it necessary? I mean, what is the broken detailed wells? That's what I mean. You have written that consume and dissipate at small length scales.
Yes.
So consumption and distribution both has to happen. That is how.
Okay. So was detailed balance fine with everybody or not fine with everybody? Not fine. I'm scared of you. You're taking my course as well. No, you can't. I mean, you have detailed balance and you coarse grain the equation. I'll try to derive some, and you will get some certain terms which are around. But if independent of that, is detailed balance fine with everybody or not fine?
Not fine.
Not fine. Okay, one not fine. Any people in the cloud?
Not fine.
Okay, I can't assume detailed balance on master equations. Okay. Okay. Okay, I hadn't planned for this. Maybe in the tutorial we'll work out a little bit of the master equation. I'll show you some examples of that. So basically, the idea is that a system which is not at thermodynamic equilibrium, that system will be driven out of thermodynamic equilibrium by certain conditions not being satisfied. Detailed balance is one of them. Detailed balance is basically a statement that if you look at the microscopic picture of the system and you look at the system as a Markov process, you look at how often the system makes a transition from a state A to a state B and vice versa. If these two things have the same probability currents, the system satisfies detailed balance. If they are not, then one state is more favorable compared to the other, and then it breaks detailed balance. And so therefore, a system which is driven out of equilibrium is something which typically breaks detailed balance. And so the patterns that I'm looking at are for systems which are out of thermodynamic equilibrium. So therefore, they are being fluxed with some energy input, which typically breaks detailed balance. So
in the case of sphere settling, lots of sphere settling,
sedimentation
due to gravity.
Yeah.
Still the dissipation will happen locally, right? Because
yes.
The sphere, but the
okay. Let me come in. So this was generally about detailed balance, and the thing that you asked about locally dissipating. So that was specific to condensed active matter. So active matter is a class of non-equilibrium systems where the dissipation and the consumption of energy happens at the level of the individual units, in contrast to being supplied at the boundaries of the system. Classical non-equilibrium system, take a jar of water, heat it at the boundary. So energy has to trickle into the ball. Active matter, the description is that each individual unit gets energy equally from the surroundings. So that was the context there. Now your question was about something settling down.
But in this case, even though that happens due to gravity, the energy is, it's settling just because of the gravity.
Yeah.
However, the dissipation will happen locally. There will be still.
Yes.
It's not unlike.
Is the question that has it broken detail balance?
I mean, is this there is a broken detail balance in this case or not? That's what my question is. Because still the dissipation. I mean, the driving force is from gravity, but however, the sphere is settling and dissipating. I mean, the energy is being given to the fluid locally. So dissipated there.
Okay, so detailed balance is a statistical statement where you talk about the probabilities of the system being informed in some state and the transitions from this state to that state and that state to this state. Detailed balance is a condition which relates the transition rates and the probabilities of occupation. If you do a coarse grain description, write down something like a Navier-Stokes equation. Is that at equilibrium or not at equilibrium? It's a bit difficult to answer because first of all, where are the fluctuations? So if you put in fluctuations there, and if the strength of the fluctuations satisfy so-called fluctuation dissipation theorem, then there is detailed balance, even at this level. So in the settling case, what you've done is to completely throw out the fluctuations. They're probably irrelevant at your level. Does that make sense? All right. Any other questions, comments? Fine. We'll meet up in the tutorial. Yeah.
Hi, Vijay.
So when we linearize the system,
yes,
and we find this, we do this linear
yes,
stability analysis. So in this case, we can find there's instability. It's an instability.
Yes.
But if we, but it might, can we have case where like if we take the entire nonlinear system, it,
yes,
it still might be stable, but the nonlinearities are actually stabilizing the system.
The state that you're perturbing or the state that you're going to?
Like that, like maybe like when we linearize it,
yes,
we might have some, we might get unstable states or something like that, but the entire nonlinear system might be still stable.
I mean, the nonlinearities are what will stabilize the other states of the system eventually. I mean, that's the thing that will keep it there. I mean, just think about the potential thing, right? It was basically a straight line there. It would be a linear thing. That's the approximation that you're doing there. Afterwards, the nonlinearities will come in and stabilize the system. So I don't understand. So you're asking if the system is linearly unstable, can it be non-linearly stable?
Yeah.
Is that the statement?
Yeah.
No, typically not.
If I understand his question, I think if you perturb it, can in the not 1D but higher dimension, can it go?
Oh, in a higher dimension, more than one dimension. Can it go in some other path and come back? And
I think that's.
I didn't think about higher dimensions.
Okay, that was not his question then. So we'll do the higher dimension analog. So there again, you approximate some hyperplane, right? It's basically a straight line there. And so it's the eigenvalues of the Jacobian matrix which will tell you that part of this. I mean, there is a caveat about some properties of the matrix. There has to be a normal matrix, and otherwise linear stability is a bit dicey. But I won't go into those aspects at all.
Okay, maybe last question online. Is detailed balance the same as time reversal symmetry?
Yes. Statistical remnant, the time reversal symmetry.
Okay, I think maybe since we have to start again at 2 o'clock, we should stop. Thanks.
Some feedback is useful. So either email me, or if you don't want to tell me, tell Abhishek and Sanjib. This guy's too fast, this guy's too slow, whatever.
So we meet again at 2 p.m. So in 1 hour, 20 minutes.